Hello, I am OnishaThapa, a graduate student in the Department of Chemistry. In this audiovisual material, we will discuss an overview of protein folding and the consequences of protein misfolding in humans. We often notice differences among people and wonder why they exist. The answer lies in genetics. Our genes code for proteins that shape the physical and physiological traits of all living things. When we look into the microscopic world of living organisms, we encounter the fundamental building block, the cell. Each cell contains a nucleus, which houses 23 pairs of chromosomes in humans. These chromosomes are made of a double-stranded helical molecule called DNA. DNA stores all the genetic codes that determine an organism's physical and behavioral characteristics. The process of gene expression is explained by the central dogma of molecular biology. In this process, a single-stranded molecule called RNA is synthesized by copying genetic codes from DNA. This RNA is then transported into the cytoplasm where it binds to ribosomes. In the cytoplasm, 20 different amino acids are available. Each group of 3 RNA bases called codons corresponds to a specific amino acid. These amino acids are brought to the ribosome by tRNA molecules and joined in a specific sequence to form a polypeptide chain. So once polypeptide chain is formed, this long chain of amino acids begins to fold into a specific 3D structure. This process is known as protein folding. Once folded right, the native conformation, the protein's perfect shape. The shape it adopts is not random. It is dictated by the amino acid sequence itself. Check out human insulin, myoglobin from a sperm whale, and human growth hormone. Each has a unique twist and functions in organisms. Ionic bonds, hydrogen bonds, disulfide linkages, and hydrophobic interactions work together like a team building a perfect puzzle. This keeps the protein strong and ready to work. So friends, now let's talk about the functions of correctly folded proteins. Proteins like elastin, collagen, and keratin are our body's builder. They give strength to skin, nails, bones, tendons, and hair. Actin and myosin proteins, they are the muscle movers, helping us flex and move every day. Meet rhodopsin and iodopsin proteins. These proteins are eye superheroes that helps us see by catching light. Antibody immunoglobulin proteins are our defenders. They fight off germs to keep us healthy. Insulin protein is a hormone that controls blood sugar, keeping our energy balanced. Here is the pepsin protein, which is a digester. It breaks down proteins in our stomach to help us eat. Last but not least, hemoglobin protein. It carries oxygen from our lungs to every part of our body. Protein folding is essential because a protein's shape determines its function. When a protein folds into its 3D structure, like a fibrous protein for structure or a globular one for reactions, it forms an active site. This site let it do specific jobs such as enzymes speeding up reactions or hemoglobin carrying oxygen in our blood. Well, protein folding often starts as soon as the protein chain is made at the ribosome. This chain, called a polypeptide, folds spontaneously because of interaction between its amino acids. Some amino acids are attracted to water, while others avoid it, guiding the chain into a specific 3D shape that's stable and functional. Sometimes proteins need extra help to fold, and that's where chaperones come in. Chaperones are special proteins that act like helpers in the cell. They bind to unfold or misfolded proteins, guide them to fold correctly, and even prevent them from sticking together in clumps. Chaperones are crucial for keeping proteins working properly. Proteins need the right environment to fold properly. Inside our cells, factors like pH, temperature, and even stress levels create the conditions for folding. For example, a slightly acidic or basic pH can change how proteins parts interact, while the cell's temperature around 37 degrees centigrade in our bodies helps them form their correct shape. Protein misfolding. Protein misfolding is when a protein fails to achieve or maintain its proper three-dimensional structure, which is essential for its function in the body. The correct shape includes an active site, a region where the protein performs its role. But in misfolded proteins, the active site may not form, which makes the protein non-functional. Misfolded proteins can accumulate inside cells through a process called deposition, where they form clumps or aggregates. These clumps, often made of sticky, misfolded protein structure, can disrupt normal cell functions such as communication between cells or energy production in mitochondria. Additionally, misfolded proteins can become toxic. When they accumulate, they trigger stress responses in the cell or form harmful aggregates, leading to degeneration and even cell death. Protein misfolding can start with a gene mutation. A mutation changes the DNA sequence, altering the order of amino acids in the polypeptide chain. This disrupts the protein's ability to fold correctly, leading to a misfolded protein. Also, defective chaperones can cause misfolding. Normally, as we discussed earlier, chaperones assist proteins in folding or refolding the misfolded ones. But if they are faulty, they cannot correct misfolded proteins, which leads to more misfolded proteins or protein aggregates. When too many proteins are made, the limited number of chaperones in the cell also cannot handle the excess. This overwhelms the folding process, causing some proteins to misfold, which may result in aggregates that disrupts cell activities. Physical or chemical stresses like heat changes, pH shifts, or toxic chemicals alters the cell's environment, and this disrupts the delicate balance needed for folding, causing proteins to misfold. So misfolded proteins are troublemakers, causing disease like Alzheimer's, Parkinson's, and many more. From amyloid beta to tau, these raw proteins misfold and clump, leading to issues causing Alzheimer's disease. Parkinson's has alpha-synuclein clumps, causing issues, and ALS has TDP43 issues. Let's explain this with Alzheimer as an example. Alzheimer is a neurodegenerative disorder from misfolded proteins. Symptoms include memory loss, confusion, and mood swings, etc. It is caused by misfolded amyloid beta and tau proteins, turning your healthy brain into a puzzle with missing pieces. In Alzheimer's, misfolded amyloid beta forms sticky plaques and tau creates tangled knots in neurons. These clumps shrink the brain and kills cell, causing memory loss, confusion, and cognitive decline. This much for today's video. Thank you so much for watching. Thank you.